Hi all. Chapter 10, Meiosis and Sexual Reproduction. Our goal here is to describe the main events of meiosis and relate those events to gamete formation. Now, meiosis is a special type of cell division. It's used only for sexual reproduction. Now, the important thing about it is that it cuts the chromosome number in half prior to fertilization. So the parents are diploid, 2 copies of every chromosome. And going through meiosis produces haploid gametes, so the sperm and egg only have 1 copy of every chromosome. The gametes fuse in fertilization to form a diploid zygote, and the zygote becomes the next diploid generation. Happy, healthy diploid generation. So this is the visual for it. We've got green chromosomes from mom, purple chromosomes from dad, and they fuse, so we've got 3 green chromosomes and 3 purple chromosomes in the offspring. Okay. Now, meiosis and fertilization can constitute a complete cycle of sexual reproduction. A complete sexually reproductive cycle. Okay. So, somatic cells, those are the body cells. They have 2 sets of chromosomes that makes them diploid. Gametes only have 1 set, so they're haploid. Now, not everything does meiosis. There are some organisms that reproduce by mitosis, like we talked about in the last chapter. They do not involve gametes. This is asexual reproduction. And an example is binary fission, like we talked about in the last chapter. Other organisms kind of can have it both ways. They can reproduce sexually and asexually. Strawberry plants, they flower. That's sexual reproduction. There are seeds. You plant the seeds, you get more strawberry plants. The other thing it does is send out runners or stolons that, as they go out, periodically they'll put down roots and send up leaves, and that's a whole new plantlet. If you cut the stolon, if you cut the runner, the little plantlet couldn't care less because it's independent. It's a whole nother individual. Okay. Even within sexually reproducing organisms, not every life cycle is the same. Okay, there are 3 types. There's most protists. We'll talk about protists in 102, but most protists have what we call a haploid life cycle, where the adults are haploid. They undergo mitosis to produce the gametes. The gametes fuse and produce a diploid zygote, which immediately undergoes meiosis to cut the chromosome number in half. So then you have haploid individuals that grow up to be haploid adults. Now, most animals have a diploid life cycle, where you have a diploid adult who has specialized cells that undergo meiosis to form gametes, which fuse at fertilization to make a diploid zygote that grows up through mitosis to become a diploid adult. Now, plants are a little weird. They have what's called alternation of generations, where you start with a diploid adult that undergoes meiosis with specialized cells to create haploid spores, which undergo mitosis to form a multicellular haploid adult. And our haploid adult undergoes mitosis to make haploid gametes, which fuse at fertilization to become a zygote, diploid, which grows up to be a diploid adult. A little weird. Or not weird, just not what we do. You'll actually spend a lot of time talking about alternation of generations in 102. We're not really going to waste a lot of time talking about it here. Okay, so mostly we're going to focus on our cells. Okay, so the sexual life cycle in animals. The cells that are going to undergo meiosis to become gametes, they're set aside really early on in development for the purpose of reproduction. Now, these cells that get set aside, they're called germline cells. They are diploid. Only these cells are going to undergo meiosis to make gametes. Okay, so we've spent a lot of time talking about homologs. This is where it becomes relevant. Okay, in diploid body cells, homologous chromosomes, well, chromosomes occur in pairs. These are the homologs. You can identify homologs because they're the same length, they have their centromeres in the same position, they have the same staining pattern when you stain them. Now, your homologs, when you look at them, one came from mom, one came from dad. And they have information about the same genes on them in the same place on their corresponding structures. So here's a karyotype. So at the bottom there you can see the homologs, the unduplicated homologs on the sides, then the duplicated homologs in the center. And you can see each of the duplicated chromosomes have two sister chromatids. Also note the non-sister chromatids there labeled, because chromatids on homologs, they are not identical. They will frequently have different information on them. Okay, this is relevant in this chapter, so keep that in mind. Okay, so homologs, same genes controlling the same trait in the same place. One copy from mom, one copy from dad, and each gene, most genes occur in a few different forms in a large population, and the variants, we call them alleles. Now your homologs may have the exact same information. You might have identical alleles, or they might be different. Now if they're identical, we say that you're homozygous for that trait. So if you got a blonde gene from mom and a blonde gene from dad, then you're homozygous for your hair color gene, and you are blonde. If you got different information, different alleles, then you are heterozygous for the trait. So if mom gave you a blonde gene and dad gave you a dark-haired gene, you're heterozygous for hair color. Okay, so meiosis. Meiosis involves two divisions. There's Meiosis I and Meiosis II, and we're going to spend most of our time talking about Meiosis I because that's really the exciting half of it. Meiosis I, what it does is it separates the homologs into two different daughter cells. That's where we go from a diploid cell to two haploid cells. Okay, key thing, we only copy the DNA before Meiosis I. We do not copy it before Meiosis I and then copy it again before Meiosis II, just the once. Okay, so Meiosis I separates out the homologs. Meiosis II separates out the replicate sister chromatids. So when meiosis is completed, what you get is that single germline cell that you started with, that one diploid cell, is now 4 haploid cells. Okay, so let's look at this. We've got this, our parent cell, our germline cell, is diploid. It's got 2 copies each of 2 different chromosomes. There's a big chromosome and a little chromosome. Let's say the red ones are from mom, the blue ones are from dad. So we get a big and a little from mom and dad. Okay. So this is, okay, keep in mind, we duplicate the centrioles when we also duplicate the chromosomes. Okay, so what happens is in meiosis I, instead of during metaphase where the chromosomes line up single file in mitosis, here they line up with the homologs side by side. And at the end of metaphase, what we have is in the middle there. We have each daughter cell having a big chromosome and a little chromosome. Okay? Whereas our parent cell had 2 big, 2 little. Now we have 1 big, 1 little. So one of the big homologous pair and one of the little homologous pair. Now meiosis II is where we pull apart the sister chromatids. At the end of meiosis I, notice that each of these chromosomes is still duplicated. And meiosis II is when we pull apart the sister chromatids. So now we have 4 haploid daughter cells. Okay. So we're going to spend most of our time talking about meiosis I. It's traditionally divided into 4 stages. There's prophase I. Key thing about prophase I is the homologs pair up and they trade parts. We'll talk about that in a little bit. Metaphase I. The homologs line up side by side on that metaphase plate. Anaphase I, the homologs separate and move to the opposite poles. Okay, so telophase 1, we've got 2 haploid cells, and the chromosomes gather at each of the 2 poles. Okay. So the key thing about meiosis is, well, to make haploid cells, but also to introduce genetic variation. Okay, during prophase 1, homologous chromosomes line up side by side, and what they do is they do crossing over, where the chromatids, non-sister chromatids, this is key, non-sister chromatids break in the same place, and pieces of the homologs are swapped. Okay, so you get a hybrid chromosome. Hang on, I got a picture of that. And this happens at a stage called synapsis, where a nucleoprotein lattice shows up between the homologs. We call this lattice the synaptonemal complex, and it looks like a ladder. Its job is to hold the homologs together and line them up so that when they break at the same point, they break, well, when they break at the exact same point. So we don't wind up with a homolog that has 2 copies of a given gene, whereas its partner, its homologous partner, doesn't have any copy of that gene. So here's crossing over, and you can see the purple and the green, they overlap the non-sister chromatids, and when they pull apart at metaphase 1, they each take a piece of the other homolog with it. And then at the bottom here, that's the synaptonemal complex, and you can see the lattice between. Excuse me. Okay, another view, this has an electron micrograph, so you can actually see it really does look like that. Now the end result is, at the end of meiosis I, we've got our two daughter cells with the homologs. Each one has 1 homolog. Now, when you look over picture D on this slide, you'll notice that our 4 chromatids are not identical. We have 1 that's all mom, 1 that's all dad, and 2 that are half mom and half dad, but are opposite in what parts are mom and what parts are dad. Okay. So the key to this is that the sister chromatids don't separate, do the crossing over. Okay. And this happens because when they exchange segments, the 2 homologs are tied together by strands of DNA. The kinetochore microtubules can gain access to only one side of each homolog because the other kinetochores are butted up side by side because of synapsis. Okay, watch this happen.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that 1 member of each pair of homologous chromosomes came from 1 parent. The other member came from the other parent. In this example, 1 parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Now there are other ways we can create genetic variation. Okay? For example, on. Next. It's having a little crisis. Okay. Now, during metaphase 1, the way the homologs pair up is a matter of chance, whether they pair up with the maternal homolog pointing to a given pole or the paternal homolog, and each set of homologs lines up independently of all the others. Okay, we call this independent assortment. And what you get is, for a given organism, the possibility of 2, because there are 2 copies of every chromosome, to the nth power. N refers to the number of different types of chromosomes. So for us, it's 2 to the 23rd power. We have 46 chromosomes, 23 types, 2 copies of each. So 2 to the 23rd gives us almost 8.4 million variations of gametes. Pretty cool, huh? But wait, we're not done. Oh, this is a visual of independent assortment, and here is a video. You can actually see how it works. This shows how having 3 different pairs will give you 8 different types of gametes. Just watch.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Okay, it's still having a little bit of an issue. Okay, but we're not done, because since we're sexually reproducing, that means that there are 2 parents for every reproductive event. Okay, when gametes fuse, the chromosomes are combined from the parents. So in humans, we've got 2 to the 23rd power. That's independent assortment from each parent. But then you have to square that because you're dealing with 2 parents. So you get nearly 70.4 trillion chromosomally different zygotes possible. Now let's say hypothetically crossing over occurs once during each reproductive event. That gives you 4 different chromatids to the 23rd power, and then square it because there are 2 different parents. And that's almost 5 octillion genetically different zygotes possible. Now here's the thing, crossing over doesn't occur just once. It occurs several times in each chromosome in each reproductive event. In other words, the number of given offspring from a given set of parents was virtually limitless. Okay, now as far as what the significance of this is, well, organisms that reproduce asexually, they produce offspring exactly like themselves, which is great when the environment is stable. But I mean, look around, the environment's never stable. So that gives us as sexually reproducing organisms an advantage, having a whole lot of variation. Because when the environment changes, and it will, the variability introduced is an advantage, because no matter what the environment throws at you, there are some individuals in the population that are bound to be able to survive because of new and different combinations of genes floating around. Now, on a less depressing level, that's also why we all kind of look like our parents and kind of look like neither of our parents. Okay, so let's talk about the phases of meiosis one at a time. Okay, the italics are the stuff that's new. Okay, so in prophase 1, we already know about how the spindle forms, the nuclear envelope fragments, the nucleolus disappears, and the chromatids, the chromosomes are duplicated. Here's where it's new. Okay, the homologs pair up. We have synapsis happening, and crossing over happens. Okay, you can either call them a bivalent, if you're more comfortable thinking of it as 2 homologs, or tetrad, if you're more comfortable thinking about 4 chromatids. Doesn't matter which word you like, they mean the same thing, so it's just a matter of personal preference. In metaphase I, instead of the chromosomes lining up single file at the metaphase plate, the homologs line up side by side. And the homologs line up, each set of homologs lines up independently of all the other pairs. So you get kind of a random mix heading for each daughter cell. Okay. In anaphase I, the homologs separate from each other. The sister chromatids do not separate, so your chromosomes are still duplicated. And telophase I, the daughter cells have 1 duplicated chromosome from each set of homologs. So they're haploid, but the chromosomes are still duplicated. Okay, so here's a visual of that. And in the photograph here of prophase, you can really see the crossing over. Metaphase, you can see them lined up single file, or no, not really single file, side by side. Anaphase, you can see telophase pretty clear. Okay, when we finish meiosis I, we have interkinesis. Okay, interkinesis is a lot like interphase, except it's usually shorter. We have less work to do here, and DNA replication does not occur. Does it occur? No, it does not. That's why interkinesis is shorter than interphase. It can be. There's less to do. Okay, so here's meiosis I in action. You can actually see it happen.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase I of meiosis I begins with the condensation of chromosomes and the vesiculation of the nuclear membrane. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase I, the spindle apparatus continues to form, and bivalents become attached to kinetochore microtubules. 1 pair of sister chromatids is connected via kinetochore microtubules to 1 pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase I. The chromosomes align at the metaphase plate during metaphase I. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase I involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in 2 cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis I, and after a period of interkinesis, the 2 daughter cells start the 2nd cell division cycle called meiosis II.
Okay, so that's meiosis I. Meiosis II we're actually going to slam through because meiosis II is virtually indistinguishable from mitosis of 2 different haploid cells. Okay, prophase II, the chromosomes condense again, if they ever decondensed. Okay, the spindle apparatus starts forming again, the nucleus disappears if it ever reappeared. Okay. In metaphase II, the chromosomes align single file at the metaphase plate. In anaphase II, the centromeres dissolve and the sister chromatids finally separate. Now they're daughter chromosomes. Telophase II and cytokinesis run through just like in mitosis. At the end, we have 4 haploid cells, and they are all genetically unique, completely distinct from one another. Okay. Here's a video, or not a video, here's a set of pictures, and you can see that the prophase II, we see the nucleus get gritty again. Metaphase II, we've got the single file anaphase. You can see the chromosomes being pulled away. In telophase II, you can see the nuclear membrane starting to reform. Here's Meiosis II in action.
At prophase II, the chromosomes condense again, and the nuclear membrane vesiculates. During prometaphase II, the spindle apparatus forms, and the sister chromatids become attached to kinetochore microtubules. In this case, a pair of sister chromatids is attached to both poles, not just one pole as in prometaphase I. At metaphase II, the sister chromatids are aligned along the metaphase plate, with each chromatid being attached to one pole. The sister chromatids separate or disjoin and begin moving to the poles of the cells during anaphase II. During telophase II, the nuclear membrane reforms and chromosomes decondense. In animal cells, a cleavage furrow is formed that causes cytokinesis or cellular division. As cytokinesis is completed, meiosis II results in the formation of 4 haploid cells. Thus, meiosis begins with a diploid mother cell that has replicated its chromosomes and ends with 4 haploid cells containing one set of chromosomes each.
Okay, and to be honest, I was just going to read this slide off, so you might as well just, oh no, go back. You might as well just read it off, pause it and read it. Okay. Now, if you compare meiosis and mitosis side by side, you can really see where they diverge. Okay. Here's meiosis I compared to mitosis again. Feel free to pause it and read it off. Meiosis II. Now notice there's a very, very little difference here. The main difference is haploid versus diploid and 4 cells that are unique versus 2 cells that are identical. Okay, here's a video comparing them side by side in action. And I've lost my cursor. There it is.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Okay, so because we love ourselves, we're the coolest animals out there, we're just going to focus entirely on the human life cycle. Okay. We go through meiosis using germline cells, so we produce a sperm and an egg that fuse at fertilization that results in a zygote. That is the single-celled stage where we all started. Okay. The zygote undergoes mitosis to grow, to develop, to become an adult. All growth occurs as mitotic division. And as a result of that, every cell not only has the same number of cells as the zygote, or I'm sorry, not only does it have the same number of chromosomes as the zygote, it has the exact same chromosomes. Okay, every cell in your body has the exact same genetic makeup, which is why we were able to take a mammary cell and make a sheep out of it. And then Dolly the sheep shows up. Okay. So that's actually something very cool. Now the differences between the cells is that each one is programmed to be a particular type of cell, but it has the genetic capacity to be anything. Okay, so here's our life cycle. You have diploid adults that have germline cells that go through meiosis to form a sperm cell and an egg cell. These fuse at fertilization to create a diploid zygote that undergoes mitosis to become a baby, that undergoes more mitosis to become an adult. Okay. So specifically, we talked about generic meiosis, but gametogenesis works a little bit differently. Okay, to form sperm cells, that's spermatogenesis, and we start with a diploid germ cell. That's the primary spermatocyte. Okay. Now, it'll go through mitosis, and one of the daughter cells will remain diploid. The other one will undergo meiosis. Okay. At the end of Meiosis I, the cells, these are secondary spermatocytes at this point, and they are haploid. Goes through Meiosis II, now we have 4 spermatids. Genetically, these are ready to go, but they need to mature and go through some metamorphosis. Okay, take a look at these things. They're built for speed. Okay, they have absolutely no extra luggage. So they have to offload their cytoplasm, get a really nice strong tail. Okay, at this point, after going through maturation, now they're ready to go. And you wind up with 4 haploid sperm. Fairly straightforward, except for the metamorphosis and maturation bit. In females, oogenesis is a bit more complicated. Okay, we start with the primary oocyte, and before birth, Meiosis I happens. And because if you look over at the sperm cell, it's carrying nothing. All it carries is its half of the DNA. So that means the egg cell has to provide everything. So when Meiosis I ends, we'll wind up with a secondary oocyte and a polar body. The polar body is that little dot there. It has no cytoplasm. Its entire function is to be a dumping ground for half of the chromosomes. Okay. Now, Meiosis II begins, or actually, Meiosis I is done before birth, and the secondary oocytes just kind of hang out until puberty hits, and then Meiosis II begins, but it does not finish. Okay, it is not triggered to finish until fertilization happens. The moment that the sperm cell gets through the plasma membrane, it triggers the completion of Meiosis II. So at this point, we now have an egg and another polar body, because the egg gets everything. The first polar body may or may not also split, so you have between 2 and 3 polar bodies. It's just a dumping ground. Now, once the sperm cell reaches the nucleus, you have fusion, and now you of the nuclei, and now you have a zygote. So what this means is you have an egg for the span of time from that the sperm gets through the plasma membrane until it gets to the nucleus. Very, very short-lived thing. Okay. I already described this, so we don't have to worry about watching the video. And if you want to, you can pull it up on your YouTube. Okay. Now, if all goes according to plan, what you have is euploidy, where you have the correct number of chromosomes in a species. But it doesn't always happen that way. Sometimes things go wrong, and that's how you get aneuploidy. Okay, this results from non-disjunction. Non-disjunction is when the chromosomes fail to divvy up correctly. So what happens is you can either wind up with monosomy, where instead of having 2 of a particular type of chromosome, you have 1, or trisomy. Instead of having 2, you have 3. Okay. There are 2 different types of non-disjunction. There's primary non-disjunction over here on the right, where Meiosis I fails to do what it should. Okay, so one of the daughter cells gets both homologs, and the other one gets nothing. And the one that got nothing, well, it doesn't have anything after Meiosis II either, and upon fertilization, the only copy of that particular chromosome was the one provided by the other parent. Okay, we show non-disjunction happening in the egg cell, but it's just as likely to happen in the sperm. Okay, meanwhile, the daughter cell that got both homologs, at the end of Meiosis II, well, each daughter cell has one of the chromatids from both homologs. So upon fertilization, you have 3 copies of that chromosome, which is not really awesome. Okay, with primary non-disjunction, there is no opportunity for a normal, healthy kid. Now, secondary non-disjunction is what happens when Meiosis I goes just fine, the homologs split up. Chances are secondary non-disjunction doesn't happen in both daughter cells. You have one daughter cell that, the sister chromatids divvy up, and so you wind up with a 50% chance of having a normal kid. Okay. Meanwhile, this other daughter cell, the sister chromatids do not split up. The spindle apparatus does not attach like it should, and the sister chromatids stay together. So at the end of the day, from this daughter cell, you have one trisomic and one monosomic. Okay. And monosomy is not at all tolerated. That's almost always an automatic miscarriage. And trisomy is slightly better tolerated. But there are only 3 types of trisomy that's tolerated beyond birth. There's trisomy 13, where you have 3 copies of chromosome 13, trisomy 18, where you have 3 copies of chromosome 18, and trisomy 21, 3 copies of chromosome 21. Trisomy 21 is the only one that's tolerated beyond early childhood. People with trisomy 21 live to be adults. Now, you all are fairly familiar with trisomy 21 because it's better known as Down syndrome. Okay, and it comes with a very distinct set of characteristics. They tend to be short. They tend to have an epicanthic fold at the inner corners of their eyes. Their faces tend to be fairly wide and kind of flat. They also tend to have a very thick, fissured tongue. It makes speaking a little difficult. It's hard to speak clearly when your tongue is thick. There's also varying levels of mental disability. Okay. Now they live into adulthood, but they tend to not live to be old because, along with the the more cosmetic characteristics, they also have weaker aortas, higher incidence of heart disease, higher incidence of leukemia, higher incidence of Alzheimer's disease. So, but they do live to into their 50s. Okay, now that's autosomal non-disjunction. Sex chromosome non-disjunction is far, far better tolerated. Then that comes about from just having too many or too few X or Y chromosomes. Okay, you can't have just a Y chromosome. There are genes on the X that are absolutely necessary to be able to live. So you will never see a YO person. However, you can survive just fine with a single X chromosome. What you get is a female. Now she's short, she's got a broad chest, normal intelligence, function with hormone therapy. A lot of these people have hormone therapy and donated eggs, and they go on to have kids. They are slow to learn, but they are not mentally disabled. They just are slow to pick up new things. Now on the left, this is the picture given by the textbook, but it does not have the really distinctive characteristics, so I grabbed the picture on the right off of the internet. you can see that really distinctive neck webbing. Okay, that skin folds on the sides of the neck. Generally speaking, though, because I mean, honestly, that picture is about as serious as it gets. You usually don't see it that pronounced. So for the most part, you would not peg these people walking down the street. I mean, they're short and kind of broad. Okay. Now, along with having one too few X chromosomes, there's such a thing as having one too many. Now, if you are male and have one too many, that gives you Klinefelter syndrome, where you're XXY. If people are male, they have underdeveloped testes and prostate. Sometimes they have some breast overdevelopment. Fairly short, long arms and legs, large hands. A lot of the secondary sex characteristics for men are missing. They don't tend to grow chest hair. They don't go bald. They don't grow a really good beard. They have near normal intelligence unless they have more than one extra X chromosome. If you have more than one extra X chromosome, that will give you some really profound mental disability. Really profound mental disability. Okay. Now, no matter how many X chromosomes are present, if there's a Y, that renders the individual male. So if you are a female with an extra X, so poly X, super female, triple X, you're female, well, because there's no Y. These people are tall, they're thin, they have some delayed motor and language difficulties, but for the most part, nobody notices. I mean, the kid doesn't walk until a little bit late. She doesn't talk until a little bit late. This individual in the picture on the right, she's got poly X. The other three are completely standard double X females. There are some theories, though, that the supermodel industry has more than its normal ratio of poly X individuals. Most of these women do not know. Okay. Now, you can have too many X's, you can have too few X's. You can also have too many Y's. That's super male, Jacob syndrome, XYY. These people, these guys, they're tall, they have a tendency towards acne and some speech and reading problems. But again, I mean, look at this guy, he's completely normal looking. I mean, you would not notice these people as being genetically different if you encountered them on the street. I mean, how many tall guys with acne do you encounter on the street? Now, I dug this one up just a couple of weeks ago, XXYY syndrome. These people are profoundly disabled. There are developmental delays, learning and intellectual disabilities, life skill disabilities, neurodevelopmental disorders, behavioral problems, which are probably linked to the neurodevelopmental disorders. As far as physically, usually tall with scoliosis, hypotonia, that means horrible muscle tone, flat feet, that goes along with the hypotonia, tremors, seizures, and excessively bent fifth digit. So like their pinky fingers and toes tend to be very, very bent. They also have some pretty profound sexual side effects, sterility, low testosterone, and yeah, this one's a little rough to live with. Okay. Now you can have the right number of chromosomes but not have the right structure. Deletions where a piece breaks off, duplications where a piece copies twice. Well, now you're going to overdose on those genes. Inversions and translocations. I've got a video for why inversions are a problem. Translocations are what happens when non-homologs swap segments. And I've got a video to explain how all of this works. Observe the video.
A chromosomal mutation is a substantial change in the structure of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. In the chromosome, shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in two places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve two chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
Okay, now this is a video on why an inversion is a problem, because despite the fact that you still have the same, correct amount of DNA, well, you got problems. This is why.
An inversion is a reversal in the order of a segment of a chromosome. Inversions do not alter gene expression unless the breakpoints on the ends of the inversion occur within a gene. When a segment of a chromosome is inverted, it can pair in meiosis only by forming an internal loop. Although crossing over occurs in the 4-strand stage during meiosis, only 2 strands are shown for clarity. Crossing over that occurs within the inverted segment during meiosis can result in some genes being lost on one chromosome, while others are duplicated on another chromosome.
Okay. So places where we see this in autosomes, that's with the body chromosomes, here's an example. This is Williams syndrome. It's caused by the end of chromosome number 7 being snapped off. And you notice these kids look an awful lot alike. Well, they're not at all related. The similarities is due to the disorder. They've got the little turned up noses, their ears are pointed, they've got the really small chin, the really wide mouth. They've got terrible math skills, but amazingly verbal and musical skills. Now, much like the non-disjunction, alterations in chromosome structure is better tolerated in the sex chromosomes. I got 2 examples for you, but before I describe them, I want to point out that the Y chromosome makes you male. Specifically, the SRY gene on the Y chromosome makes you male. Now, if it happens to not be on the Y chromosome, if it happens to be snapped off of the Y chromosome, what you get is somebody with Swyer syndrome. Physically female, genetically male. These people find out generally when they fail to start puberty. And then they find out that, surprise, you're a guy. And it'll mess with your self-image. The other side of that coin is de La Chapelle syndrome. It shouldn't be La Chapelle, it's de La Chapelle, where that SRY gene that broke off the Y chromosome, well, when it lands on the end of the X chromosome, you wind up with someone who is genetically female, XX, but physically male. Here, let me show you some pictures. Okay, these people on the left, these are all people with Swyer syndrome, and you'll notice that they look like women. That person on the right, that's someone with de La Chapelle syndrome, and he looks like a guy. Okay, none of these people you would look at and be like, no, that's definitely not a man. It is. Now, generally, people with Swyer or de La Chapelle, upon finding out what they are genetically, they start taking hormones to kind of even them out, and choose to continue to live as the sex that they've always been. So it doesn't throw you off too much. The woman up top, the blonde, her name is Jeannie. She's the poster child for Swyer syndrome, and she's married to a man, and they have 2 adopted children. Okay, so as far as she's concerned, she's a woman, and she just goes with it. Any questions or comments? Please do contact me. Thank you, and good night.